Good morning, everyone. It's a great honor to have been asked to give this lecture, and I'd like to thank the Scientific Program Committee for choosing me. I think one can't help but wonder that the fact that the meeting is in Toronto might have influenced them a little bit, but hopefully that isn't the only reason that I was asked to give it. So as you've heard, I'm going to be speaking about the genetic locus symbols, emphasizing that we've got a broken system and it really is time to fix it. I have nothing relevant to disclose related to the lecture, except for the fact that I feel very, very honored by being asked to give a lecture in Stan Fahn's name. Stan is a friend and mentor, and it really is truly one of the greatest privileges and honors I've been given to in the request that I give this lecture. Now, Stan has established a tremendous tradition, and I'm going to use this picture from the Paris meeting that we had a couple of years ago when I was president to serve as an analogy for part of my talk, and I'm going to come back to this picture. You see the legacy that Stan has led in establishing presidents that followed him. He was the first president of the Movement Disorder Society, and I'll use this picture as a way of emphasizing some of the points I'm going to make, particularly related to missing loci or vanishing loci. And so remember this picture, and we're going to come back to it at the end. I'd like to emphasize that everything that I say this morning will relate to very hard work on the part of three collaborators, with myself serving as a rather minor role player. Connie Maris, Kaja Lohman, and Christine Klein have been instrumental in developing many of the arguments that I'm going to raise for you today. So anything you like or appreciate about what I say this morning is credit to them. Anything you don't like or anything that I make mistakes on, I'll take full credit for. The premise I'm going to present to you today is that we have very significant problems with our current locus system of designation. We have multiple sources of confusion, the system perpetuates misinformation, and it's misrepresented as a useful research or reference tool for the list of inherited disorders of particular phenotypic classes. And today I'm going to be emphasizing the phenotypic classes of dystonia and Parkinsonism. So our premise this morning is that this system is terribly broken, and it really is time to fix it. The objectives of my lecture will be to review this system, the history behind it, and the current use of the locus symbols, to discuss those problems that we've designated as important, using, as I've mentioned, dystonia and Parkinsonism as examples, and to propose some specific recommendations as to how we might revise the system. This is a very large table just to show you where we stand with the DYTs. As you can see, there are 21 of these, and there are many problems with the designation. And almost purposefully, I've listed them in a way I can't read them. I'm not sure if you can. And then, secondly, the PARC system, up to 18 of these designations with selected problems that I'm going to be drawing your attention to over the course of this lecture. Let's start with a little bit of history. The HGNC, or Hugo's Gene Nomenclature Committee, was founded in 1979. It was led by Dr. McAlpine, and now a team of postdoctoral fellows and bioinformaticians perform the work that Dr. McAlpine did. The funding comes from both the United States and the United Kingdom, and this committee has assigned unique gene and symbols for over 30,000 known human genes, many from the Human Genome Project. Now, originally, this system was intended to introduce specific chromosomal regions linked to a familial disorder where the gene had not yet been discovered. So it served as a placeholder for genes. And then once the gene was discovered, the plan is that this would be formally withdrawn and merged into the entry for the clone gene following that gene discovery. However, there are certain examples where the gene name has not been agreed to, and so, for example, in the case of PARC2 and PARC7, this remains without a gene name terminology. But the original plan was to merge and then remove the locus symbol. Now, if you want a locus symbol, you perform an online request, and you can see the points that you're requested to provide in the online form. And most of these requests come from databases and individual researchers. Now, that's the way it should be. Unfortunately, there have been a number of problems that have developed since then. So subsequent developments the system has really become entrenched in common use. It's used as a catalog of known inherited disorders, and in fact publications of novel loci are made without the request, and then HGNC has to do some catch-up to define the locus symbol in the case where it hadn't been requested. And remarkably, we're in a state now where investigators actually assign their own locus symbol without even seeking approval from HGNC. So instead of the intended use, where we're talking about a chromosomal region, for example, the Park5 locus, it's much more common that incorrect uses of the terminologies have been applied. So, for example, I've given here the gene mutation. We tested for DIT6, or as the name of the disorder, DYT1 dystonia. And indeed, dystonia 1 is a term that has crept into the literature. So, for example, in this excellent review article by Mueller in 2009 on the monogenetic primary dystonias, you see instead of DYT, dystonia 1 through 20 has been listed. And so we've got a drift of DYT to actually equilibrate or be the equivalent of dystonia now. So I'm going to draw your attention to a number of major problems that fall into these 7 categories, give you some examples, and then at the end try to recommend some solutions. The first problem is that we have a combination of risk factors and disease-causing genes in the same system. So originally, most gene loci were identified through classical linkage, and this assumed a monogenetic cause in all the members of the single family that was described. So in this a very well-known diagram from Manillo in 2008, we see what we're describing with this monogenetic form, rare alleles causing Mendelian diseases, in the top left-hand corner, where on your y-axis you have increasing effect size, and on the x-axis increasing allele frequency. The problem is now, as you know, we're dealing with complex disorders, so the common variant, common disease hypothesis has come out, and this is where common polymorphisms actually confer disease risk, in contrast to being a monogenetic cause of the disease. And this comes from, for example, linkage studies in unrelated individuals in isolated communities such as Iceland, where Park10 was discovered, and genome-wide association studies where at least 3 Park loci have been defined. So here we're dealing with examples down on the bottom right-hand corner where allele frequency is common, but the risk is, or the effect size is rather low. Now, it's important to consider the accuracy or the importance of a diagnostic test, the utility of a diagnostic test, using the area under the curve of ROC curves, receiver operating characteristic curves. And importantly, as you see here, if I get my arrow, random guessing is really only at 0.5, and you see the expectations or demands for screening individuals at risk or using a test as a pre-symptomatic diagnosis in the general population, demanding much higher areas under the curve. Now, this very important study published in The Lancet this year described an analysis of 5 very large GWAS studies, a meta-analysis, and in this they identified 11 loci that were suspected to reach or have reached the threshold for importance in causing Parkinsonism. In their meta-analysis of these 5 large GWAS, we see the area under the curve at 0.63 with the relative risk in the largest or 5th quintile of 2.51. What does that mean? Well, if we return to the ROC curves, we have an area under the curve of 0.63, which really doesn't confer any reasonable degree of discrimination between cases and controls. In fact, carrying all 11 significant associated variants increases the risk of Parkinson's disease by 2.5, and as Klein and Ziegler pointed out in their editorial, the prevalence of Parkinson's being approximately 0.14 in the general population results in a lifetime risk of developing Parkinson's with all 11 risk variants of 0.35%. So let's put that into the consideration of the relationship with monogenetic causes that we've been talking about originally. So now we're no longer talking about a single explanation, but only a very small part of the puzzle. Let's contrast 0.35% lifetime risk with 100% lifetime risk in the case of some of the monogenetic causes of Parkinson's, for example, being homozygous for PARK2 gene. Now, another point related to these risk factors is that the risk factors often have an inconsistent application of the loci designation. So in the case of glucocerebrosidase mutations for Parkinson's, we have what some people believe is the strongest risk factor for Parkinson's, and yet this hasn't been designated with a PARC. So as you see, in the case of Ashkenazi Jews, about 20% with GBA mutations, and in non-Ashkenazi Jewish individuals about 6 to 7%. So the practice of assigning risk factors as loci has really been very inconsistent. You see some of them designated, and other two very important ones that you're aware of, GBA, as I pointed out, and MAPT, discovered or confirmed in that GWAS study that I've shown you already, not designated with locus designations. Another problem is that we now recognize that a single gene can provide both a risk variant and be important in terms of positive causative mutations. So here you see SNCA, or alpha-synuclein gene, as well as LRRK2, in which case both distinct mutations cause disease, but also variants in these genes are considered risk factors for Parkinson's disease. And the problem is that the current system has no way of separating or indicating the difference between these two designations. And you can imagine, as this picture, as this map gets more and more complex, in the original figure from Manillo's article, the current system is going to fall apart even more than it is now. The second problem I'm going to point out is that there's a complex relationship between the phenotype and the list membership. So, for example, if we deal with the DYT loci, all of you are aware of the fact that some of these represent disabling generalized dystonia, for example, DYT1 and 6, but this list also includes disorders that may look nothing like that. For example, the paroxysmal dystonias are often variable dyskinesias that actually never show dystonia. So it seems rather illogical to include that in the same locus system. In the case of PARC loci, as you know, some of these disorders are clinically indistinguishable from Parkinson's disease, and others represent disorders that you'd have no trouble distinguishing, that look nothing like Parkinson's disease and actually behave in terms of their clinical course, nothing like idiopathic Parkinson's. The problem is that as an initial family is described, often the patients may be clinically atypical or not fully representative of the clinical phenotype of that disorder. And in fact, in certain cases, we recognize widely that more than one movement disorder may predominate. So for example, with Lubag and rapid onset dystonia Parkinsonism, both of these, or these disorders, both have Parkinsonism and dystonia quite characteristically. Another problem that we've pointed out or been concerned about is that in the current system, especially as individuals are assigning their own loci, there may in fact be non-scientific motives for choosing a specific PARC or DIT characterization, depending on the impact factor of the journal you want to publish in, depending on who is providing grants, and so there may be a preference to choosing, for example, a PARC loci over a DIT loci in the case of publications when people are assigning their own loci designations. A third problem is that we have some missing locus symbols. Some genetic disorders have never been granted a locus symbol. The discovery doesn't come to the attention of the HGNC, for example. More and more, we're going to find that the causative gene is discovered before linkage occurs. And this is really occurring on the basis of advances in technology. A gene discovery is coming without the need for linkage, as all of you know. An example of this I could give is Dopa-Responsive Dystonia. So we have on the left DYT5A caused by GTP cyclohydrolase 1 mutations, 5B by tyrosine hydroxylase mutations. However, we now know that sepiapterin reductase mutations cause an identical clinical phenotype, and yet this disorder has not been given a DYT designation. It's logical then that emerging techniques or technologies are going to allow as a first step the discovery of a gene without the designation of a loci. So we need a system that takes that into account and can advance and proceed as these modern advances bring gene discovery to the fore. And as I've pointed out, and as I'll continue to point out, our current system doesn't have any method of doing that. We need a system that integrates new discoveries into current nomenclature. A fourth problem is that we've got long-standing designated loci that still have yet to have an associated discovered gene. An example would be DYT13, first reported in 2011 by linkage studies in patients with cervical or upper limb, craniocervical and upper limb dystonia. And despite the 10 years that have elapsed, there have been no additional studies that have demonstrated linkage. In the case of the Park5 discovery with ubiquitin carboxy-terminal hydrolase L1 mutations, these were discovered in a single family, very exciting in terms of our potential understanding of the pathogenesis of Parkinson's. However, mutations have been found in no additional families. So one has to really question the importance of these discoveries and the relevance to designation in a firm locus system. So there's considerable uncertainty in these cases and others whether or not a locus or a gene is truly linked to the disorder. And the current system of nomenclature actually provides no way of distinguishing confirmed and unconfirmed loci like these. A fifth problem, I think, is expected given the advances in technology and older technology that's been used as locus designations have been provided, and then technical changes occur and we see these past errors fairly quickly. So it's not surprising that some of these errors will occur. So for example, in DYT14, clinical miscalculation or misclassification of a single case. For example, this case circled here had dystonia, mental retardation and seizures, but was on neuroleptics. And the LOD score changed considerably when this individual was reclassified. And then the investigators expanded the assessment and looked at gene dosage studies of known genes and discovered that, in fact, DYT14 was due to GTP cyclohydrolase 1 mutation. And so really this doesn't exist, and it should be redesignated as DYT5A. Another very well-known example to all of us is the Park4 situation where a sample handling error resulted in the designation of Park14, and then this seminal paper by Singleton and colleagues defined the presence of alpha-synuclein locus triplication in the case of Park4 and redesignated this to Park1. So there are a number of examples that we can cite. The problem is that these disorders have now been assigned a specific symbol. They're in a holding place, and this entrenches the designation in the literature, and it's very difficult to retract it given the current system. A sixth problem is that, in fact, locus symbols can exist without a locus. So a symbol has been established to indicate a family with a seemingly apparent inherited disorder without any linkage whatsoever. In 1934 and subsequently in 1988, Spanish gypsies were discovered to have a form of what was presumed to be autosomally recessive inherited dystonia, and it was subsequently designated as DYT2. And since then, there have been a small series of papers that have described similar families with what has been termed as DYT2-like disorder. The problem is that we have no idea whether these represent different genetic disorders or a single disorder, so it seems rather inappropriate to define them with this locus designation of DYT2. So it may be that this is describing, or these families are all completely different genetic disorders. Another example we might cite is DYT4, where originally this was used as sort of a mixed bag of autosomal dominant DYT1 negative primary dystonias. This has been subsequently reapplied to a single family with what was described in JNNP in 1985 as hereditary whispering dysphonia. I think this is a fairly useful designation if we're doing it only to draw attention to investigators that might have similar families. However, it does seem inappropriate or not terribly sensible to take these observations and ingrain them into a system of locus symbols. We need a way of drawing attention to investigators of these types of families, but should we really put them in a formal locus symbol list at this point? The final problem that I'm going to draw to your attention is the potential for erroneous duplication of loci. This is an important example of that is in Press in Neurology. This paper was published in, I think it's 1995, where paroxysmal choreoathetosis associated with spasticity was defined with this family as DYT9. Weber and colleagues have re-looked at this same family and have realized that the phenotype is paroxysmal exercise-induced dystonia, that there is ataxia, spasticity, and seizures present in this family, and recognizing the phenotype is rather similar to GLUT1 deficiency, they were able to define a mutation in the SLC2A1 gene, and so we now know in Press, as I say, that DYT9 should really be the same as DYT18, or GLUT1 deficiency. And if we look at some of the other paroxysmal dyskinesias or paroxysmal dystonias, DYT10 and 19 and DYT8 and 20 have similar clinical and locus overlap, and it may be that in these we're dealing with further examples of locus duplication. So in this diagram, I'm listing all of the DYT and PARC loci in a system that starts with a locus assignation through to the final identification of a gene and the replication of the gene being associated with this disorder. So everything to the left on this diagram is confirmed. The problem is we've got a lot of the disorders that are entrenched in our locus system that are nowhere near the side of this diagram we'd like to see them on. So we have major problems. We have a system that combines disease-determining genes with risk factors. We have inappropriate and erroneous symbols, missing loci, and sometimes weak relationships between the membership, list membership and the phenotype of the disorder we're describing. And the consequences of all of these problems is that we're dealing with an incomplete list in case of PARC or DIT of disease-associated loci with many inaccuracies, and often the relationship that exists between the locus and the disease range from actually non-existent through to only risk-confirming all the way down to what we would really like, disease-determining. And so we've got confusion, perpetuation of misinformation, and this all ends up causing misrepresented, a system that is misrepresented as a useful reference tool. So the question now that I'm going to pose in the last part of the lecture is, we've got a broken system. I hope I've convinced you. Let's figure out how we can fix it. And so the royal we, not me, the four of us that I've mentioned at the beginning of my talk, are going to make a series of recommendations that I'll cover rather quickly. The first problem, as I've mentioned, the current numbering system conveys no useful information, we believe, at all. We have locus misassignments as well as no assignment at all. We have the potential for non-scientific motivation promoting the inappropriate assignment of loci. And just as an example, we have, in my early slide, 21 DYT assigned, only 13 actually exist. So our recommendations, we're suggesting that a symbol prefix be followed by either the gene name or the chromosomal region. So when we finally have a gene name, the chromosomal region would be replaced. And you see here a couple of examples of that. So this approach would convey whether or not the responsible gene has been defined. And we strongly believe, given the use of these systems by clinicians as well as researchers, that we need to maintain a connection between the phenotype and the gene, rather than allow HGNC to reassign the gene locus and then remove the assignation from the system entirely. I think it's really very important that we have a usable system that clinicians can find valuable to them. We're not the only ones proposing this approach. Jackin and his colleagues recently suggested a very similar approach to nomenclature of congenital disorders of glycosylation, and I've included only one of 4 tables in that report for your interest. The next problem we have is the fact that our current system combines disease-determining genes with risk factors. And we feel that risk factors would never be used to diagnose a condition. An example of how this might be inappropriate is the fact that environmental risk factors would never be incorporated into a disease classification system. So we would recommend that we maintain two completely separate lists, one of disease-causing mutations and the other of risk factors. In fact, there is already an excellent genetic risk factor catalog in PD-Gene, and we'd encourage the continued use of that. And when we have a system or a situation where we have a disease-causing mutation and a risk factor in the same gene, it's reasonable to include that in both lists, in both the risk factor catalog as well as the symbol list. The next problem, the multiple inaccuracies and redundancies of the current list of locus symbols. I think this could have been avoided to a great extent if there was a higher level of evidence that was required prior to assignment of a symbol. And it doesn't seem appropriate that the master list that we all use to characterize genetic disorders be a place for preliminary findings. So we feel that we need to raise the threshold of evidence before locus assignment is provided. And we propose that maybe committees be developed, for example, established by the Movement Disorders Society, where experts provide recommendations. We've provided a couple here. So, for example, you need more than one independently ascertained family with linkage, or at least two families with a similar phenotype and a genetic mutation before you allow them to be entered into the locus list. The next problem is the complex relationship between phenotype and list membership. The locus assignment is sometimes done before the full spectrum of the disorder is known, and so we need a system that can evolve and change as the disorder is better recognized and understood. So as I pointed out earlier, there are mixed patterns for diseases that are very common. For example, dystonia and Parkinsonism are a very common combination. And then the problem of paroxysmal dyskinesias is really a significant one that needs to be addressed. So our recommendation is we assign an appropriate phenotype prefix to relationships. Raising the threshold for assignment should help this problem. The involvement of expert reviewers in locus symbol assignment should help as well. And we're going to provide or recommend two additional points. That is, the first, if we have a disorder that commonly has two phenotypes, let's allow a double prefix. For example, DIT-PARC. XQ13.1 for Lubag, for example. And in the case of the paroxysmal dyskinesias, we feel that a unique prefix is appropriate. It's inappropriate to maintain paroxysmal dyskinesias in the DIT locus symbol group. So this slide very briefly shows you all of the problems that I've outlined for you this morning. You can scan it yourself. We have a number of solutions that I've tried to quickly outline for you, and you can see that these problems are managed by or handled by a number of the solutions that I've outlined this morning. And just to show you the outcome that we might propose, and again, this is simply a proposal. This is a list of the genetically determined Parkinsonisms that follow the recommendations that we would make. I've divided them into autosomal dominant, X-linked, and autosomal recessive. So you see here, for example, the X-linked has a dual prefix recognizing dystonia and Parkinsonism in the X-linked dystonia Parkinsonism syndrome. So you see here where genes are known, it links to the prefix. When there is only linkage, we don't have that. In the case of dystonia, the new assignment would be autosomal dominant, X-linked, and recessive. And again, you can scan this list, and the current locus symbol is listed on your right. And then finally, we're recommending a completely separate group for the paroxysmal dyskinesias as listed here. Now, finally, it's important for us to start thinking about how we can affect this change. We're arguing that our current system really is doing a major disservice to our field, and it really is serving as a stumbling block to advancement in genetic research and clinical medicine. Now, precedents do exist for changing terminology and classification systems. I've given you one example in the case of the congenital disorders of glycosylation. And in the case of terminology, we're all aware of the move from calling Haller-Worden-Spatz disease HBIA1, given the history behind Haller-Worden and Spatz genetic experimentations. So how can we effect this change? Very briefly, this requires international collaboration. It's not going to be accomplished by me getting up and talking to you. We need to plan how to do this with input from geneticists as well as clinical experts, between nomenclature teams and clinical and research communities. I'd argue that we need to develop task forces. For example, in the case of our disorders, dystonia, Parkinsonism, for example, MDS should develop a task force that would advise on the current system and assist in designation of locus symbols. We need to involve representatives from phenotypic databases, recognizing that HGNC is there for developing a genetic database, and so we need to merge these two interests. We need to establish formal mechanisms for reviewing and revising the list by the groups that are advising on these procedures. And I think a very important point, we need to involve journal editorial boards so that new symbols can't be published without the appropriate process undertaken as outlined above. So in conclusion, I think we have a very significant problem with our current system. Significant improvements in the system are possible. We really strongly believe that, and very definitely necessary. Next generation sequencing is not going to solve the problem, it's only going to make it worse. And so a new system of nomenclature will be needed to accommodate the incredibly large amounts of accumulating new information on causative and susceptibility factors for diseases, and we need to do this in a rigorous and logical way. So coming back to the President's locus system, as I've mentioned, I'm using this as a bit of an analogy for my talk this morning with missing loci or disappearing loci. So some of you may not know that one of these presidents wasn't at that dinner, and so with the magic of modern photography, we were able to bring that missing locus back into our system. And now at this meeting, I'm very delighted to tell you that a new presidential locus has been defined, and we're now placing him into the presidential locus symbol system. Thank you very much.